Hello. Today we want to cover Chapter 12, the cell cycle, and learn about how cells live and die. Part of their cycle is what we have drawn up here at the front, where you can take cells and then they can go through a process to divide. It's only a small portion of the cell cycle, but that's what we'll be talking about in today's lecture. At the end of Chapter 12, we'll then jump into Chapter 18 to look at the cellular mechanisms behind cancer, which is related to the cell cycle system being broken. The first thing we want to talk about is the roles of cell division. There are three key roles of cell division that you should know and be very comfortable with. First, cell division is important for asexual reproduction. So if you're a single-celled organism and you're able to go through cell division, like this amoeba is over here, this is how you produce offspring, which is one of our key characteristics of life. Secondly, growth and development. All of us used to be just a single cell big. Through the process of cell division, we're now full of millions of cells. And repair. That's our third key role. We are able to repair damaged tissues through the process of cell division. So you should absolutely know these three key roles of cell division. First, it's for asexual reproduction. Second, it's for growth and development. And third, it's important for repair. Please remember that cell division is just a small part of the overall bigger picture of the cell cycle. On this slide, what I want to do is introduce some terminology that we'll be using in the upcoming chapters. Let's focus on the left-hand side of the screen first to talk about somatic cells. Somatic cells are normal body cells or non-reproductive cells. So think of like a skin cell or a liver cell or a bone cell. These cells all have two sets of chromosomes in them. So if I took out a skin cell and we looked at it in a microscope, right, we would see that I have two copies of chromosome number 1. I got one copy for my dad and I got the other copy for my mom. And that's how your normal body cells look. We call these cells diploid. That prefix di means 2. We use this formula 2N to describe these somatic body cells. You'll sometimes see it written in the following way. 2N equals 40. This is not an algebra equation. You don't need to solve for anything. The 2N part is just showing us that this cell is a diploid cell. This is a normal body cell. The second number, the 40 in this case, is telling us how many chromosomes. So as I look at that down at the bottom of the slide, 2N equals 40, what this is telling me is I'm looking at a normal diploid somatic cell, and there are 40 chromosomes inside that cell. Again, no need to solve for N in these types of instances. When somatic cells go through cell division, they go through a process called mitosis. That's what we're going to learn about in today's lecture. When we're done with the process of mitosis, we end up with two cells that are identical copies to one another and to the original cell. So that's really the focus of what we want to do today in Chapter 12. In the future, though, in Chapter 13, we're going to slightly shift gears and talk about another type of cell. So I want to introduce that. Again, this is more when we get to Chapter 13. In Chapter 13, we're going to look at gametic cells. Another name for the word gametic is to say that these are reproductive cells. So if you're a girl, we're talking about eggs. If you're a guy, we're talking about sperm. In egg and sperm, there is just a single set of chromosomes. So if we pop out one of my eggs and look at it, there's just going to be one copy of chromosome number 1. These cells are called haploid, or they're abbreviated with the letter N. So for these types of cells, you may see terminology that looks something like this, N equals 16. The N part lets us know that we're looking at a haploid or a gametic cell, and then 16 tells us that there are 16 unique chromosomes within that cell. When gametic cells go through cell division, they have to go through a special type of cell division called meiosis. Again, that's what we'll focus on in Chapter 13. Meiosis is going to result in cells that have unique characteristics one from another. So one sperm cell is not going to look like the other sperm cells. Another word for gametic cells is to say that these are germ cells, so not germ like cough, cough, I'm sick, but it's just a way that we talk about these egg and sperm. And again, specifically, gametic cells are only the egg and sperm. In Chapter 12, we want to introduce some vocabulary that we're going to use. This vocabulary is very similar. I was not the person who named them, so don't get upset at me, but we do want to be careful about these terms. So let's just go through them and make sure we're comfortable with this terminology you'll see throughout the chapter. The genome is all of your genetic information. So if we look at one cell and we focus in on the nucleus, all of that purple stuff in there is that cell's genome. Every single cell has all of the instructions for that entire organism. It might not be using all of those instructions, but that genome is that entire genetic set of information. In prokaryotes, it's going to be a single circular piece of DNA. In eukaryotes, however, that DNA is going to be broken up into multiple linear strands. And in eukaryotes, what we find is that DNA is going to be associated with a high amount of protein. So when we're looking at that purple stuff here inside the nucleus, right, we can say that that's DNA and proteins that we're visualizing right there. The DNA plus the protein can be called chromosomes, or it can be called chromatin, depending on its coiling. And we'll look at that in a moment. Every eukaryotic species has a characteristic number of chromosomes. For humans, our chromosome number is 46. So it turns out for humans that in a normal body cell, we could write the following. In a normal diploid cell, we have 46 chromosomes. That means you got 23 chromosomes from your mother and 23 chromosomes from your father. And again, the 2n part means that you have 2 of every kind of chromosome. Let's go through some of these terms and try to draw them. Chromatin is the structure that we can see when we look at a cell and we're looking in the nucleus, and the DNA and the proteins are all loosely associated with one another. So that purple gunk that I'm sort of drawing in there, that's DNA and protein. It's very loosely coiled. This is what cells look like most of the time, and we call that diffuse mass of DNA and proteins chromatin. When the cell gets ready to replicate or divide, that DNA and that protein, that chromatin, they are going to condense. So instead of being gobbledygook, we're going to start seeing very distinct structures. Those distinct structures are what we call chromosomes. There are 2 different types of chromosomes we're going to see, and I want you to be familiar with both. So first we'll start off with an unduplicated chromosome. Let me erase to give ourselves just a little bit more room to draw. An unduplicated chromosome is often drawn as a single line, or sometimes you'll see it drawn like this. That's an unduplicated chromosome. So this is a piece of DNA and protein that's tightly coiled together. When cells get ready to go through division, they're going to take their chromosomes and they're going to make copies of that information. When they do that, we end up with these things called duplicated chromosomes. You'll often see duplicated chromosomes drawn as an X, or sometimes they're drawn in more of that structure. Those 2 structures are duplicated chromosomes. On duplicated chromosomes, that X is still just counting as 1 chromosome. Yes, it's been duplicated, but that is 1 chromosome. Likewise, when you have an unduplicated chromosome, that still just counts as 1 chromosome. As we look at our duplicated chromosomes, we can identify some additional features. In our duplicated chromosome, we want to sometimes talk about 1/2 of the duplicated chromosome in relationship to the other half of the duplicated chromosome. Those are called sister chromatids. Sister chromatids only ever exist as buddies. They do not exist singularly. So if we look at our duplicated chromosome up here, this chromosome right here has 1, 2 sister chromatids. However, when we look up at this unduplicated chromosome, it is 1 chromosome with no sister chromatids. In the blobby pictures, you'll notice that I left sort of a narrow region on both of the chromosomes, sort of right here, that little narrow space, and down here, that narrow space. That narrow space is called the centromere. This is an area where the DNA and the protein stay very tightly coiled. It's a narrow waist, is what we sometimes call it, that we see on both unduplicated and duplicated chromosomes. When the cell begins to divide, what's going to happen is microtubules. Remember those from back from chapter 6. So here are some microtubules. Microtubules are going to come out and they are going to need to grab onto our duplicated chromosomes. Instead of having those kinetochore microtubules grab directly onto the purple chromosome, they're actually going to grab onto a protein called the kinetochore. So that's what I'm drawing right here in blue. This is a big kinetochore protein. Okay, so the microtubules will grow out and they'll grab onto the kinetochore. And it'll happen on the other side as well. Microtubules will grow out and they'll grab onto that kinetochore protein. Those microtubules will then be able to sort of jerk and yank around this chromosome as it needs to move throughout the cell. So that's what a kinetochore is, that sort of blue jelly bean structure we just drew. Lastly, we want to talk about a cohesin molecule. When we look at a duplicated chromosome, we have one sister chromatid and a second sister chromatid. To hold them together, there's a molecule in between them that's right here. Think of it sort of like as glue or sticky tape or something that's holding those two sister chromatids together so that we get this molecule, right? So we're talking about what's happening right here in the center that's holding those two halves together. That is a protein known as cohesin. So cohesin is the protein, think of it as something that's causing cohesiveness or cohesion, but that protein, cohesin, is holding those two sister chromatids together. Again, these are vocabulary words we'll be using throughout our discussions in chapter 12 and 13, so hopefully that helps you visualize these structures a little bit better. Here we see them all written out for us in nicer terms. So before cells divide, DNA gets replicated and chromosomes condense. You should be able to differentiate between duplicated and unduplicated chromosomes. So duplicated are the lines versus duplicated are the X's. Sister chromatids only exist as sisters. So up here at the top, we've got a picture of one duplicated chromosome, and you can see that there's one sister chromatid and a second sister chromatid. The centromeres are the narrow region where they sort of have that waist, so it's this area right here. Kinetochore proteins are where the microtubules grab on to the duplicated chromosomes. You can see it really well in this side picture. So the red parts are the spindle fibers, and then those green bits are the kinetochores. And you can see how the spindle fibers are attaching onto that kinetochore to move those chromosomes around. And then cohesin is the molecule that happens and is between the sister chromatids that's holding the two halves of the X together. As we start talking about eukaryotic cell division, we're going to see that eukaryotic cell division really takes place in two different parts. First, we're going to have mitosis. Mitosis is division of the material within the nucleus. So when we say the word mitosis, we're talking about DNA in the nucleus dividing. We're just going to focus on is what is going on in the nucleus. There's another part to cell division, though, called cytokinesis. This is interested in the rest of the cell. How do I divide up the mitochondria and the cytoplasm and the Golgi apparatus? That's called cytokinesis. So when we look at cell division, we want to remember there's two parts. Dividing the nucleus, that's mitosis, and then dividing the rest of the cell, that's cytoplasm. Here we've got a nice image. Let's see if we can recall sort of what cell division is going to look like. Up here in this top cell, you can see that we have one unduplicated chromosome, and there's a centromere. There are no sister chromatids. To get ready to go through cell division, we're first going to duplicate the DNA. So now we've duplicated the DNA. There is still just one chromosome. It's now a duplicated chromosome, and so we have twice the amount of DNA. We still just say that there's one chromosome there. We then go through the process of cell division, which is going to divide the sister chromatids, and now we end up with two daughter cells. These two daughter cells are identical, right? They look exactly the same to one another, and they look exactly like what we started off with. You might notice that I'm using the term daughter cell. This is our terminology we use. It has nothing to do with X and Y chromosomes or the sex. It's just what we call the cells that are produced through the process of cell division. So let's go ahead now and look at the details of mitotic cell division. Let's go ahead by starting looking at the cell cycle. The cell cycle consists of the mitotic phase and interphase. So the cell cycle is the entire life and death of a cell. So if we think about the cell cycle, it's got a big portion which is going to be interphase, and then a smaller portion, which is going to be the M phase. So most cells, they start off their life right here, and then they're just going to sort of start progressing around, and they're going to continue around this cycle throughout their life. The big part of the cycle is called interphase. This is where most cells are most of the time. This is where there's going to be cell growth and copying of chromosomes. This smaller wedge, mitotic phase, is going to be where we do both mitosis, that's division of the division of the nucleus, and then cytokinesis, which is going to be division of the cytoplasm. Interphase is where we spend about 90% of our cell cycle. So again, we're starting off our life as a brand new cell right here at the beginning of interphase. The first part of interphase is called the G1 phase, or the first gap. This is where the cell is growing and just doing its normal job. After G1 comes the S phase. S phase stands for synthesis, because this is where the DNA is going to make copy of itself. This is where the cell has recognized that it's going to need to divide, and it's making a copy of its genetic information. After S phase, we then find ourselves in the G2, or the second gap phase. This is where we're going to start building some of the machinery needed in order for the cell to go through cell division. They're going to duplicate the centrosome region. So that entire bluish part right here, that's all interphase. This smaller element down here at the bottom, this is the mitotic phase. You can see that the mitotic phase is going to be composed of mitosis, which is dividing the nucleus, and cytokinesis, which is dividing the cytoplasm. I wish this picture had a little bit more detail for you about mitosis. So in mitosis, we can actually take mitosis and divide it into 5 phases. I often just call them by their initials and call it PPMAT, but you should understand what PPMAT stands for in mitosis. So here we go, PPMAT. In mitosis, when this nucleus is dividing, the first thing that happens in prophase is the chromosomes become visible. What's happened is during interphase, as you look at the cell, the DNA is all in chromatin form. So this is what the cell looks like in G1. When the cell gets to S phase, the chromatin's there, and then we duplicate it. But again, it's just in chromatin form. When we get to G2, again, the DNA is still just in chromatin form. What happens when we get to prophase is now when we look at that cell, we're going to start to see duplicated chromosomes appearing. In prometaphase, nuclear envelope is going to disappear. So we're going to lose that nuclear envelope, and spindle fibers are going to begin to come and attach to our chromosomes. In metaphase, this is going to continue until all of your chromosomes have lined up across the middle of the cell. In anaphase, those chromosomes are going to begin to pull apart, but we want to make sure that we're going to use the right terminology. We're going to say that chromatids, specifically sister chromatids, begin to pull apart. Down here at the bottom, you can see me sort of sketching this out. The sister chromatids begin getting drawn to either side of the cell. So in anaphase, things are pulling apart. Think A for apart. And then we get to telophase. In telophase, what happens is we're just focused on the nucleus. The nuclear envelope is going to reform, and the DNA on either side is going to go back into chromatin form. So mitosis, those beautiful black pictures we just drew around the edges of the screen, is what's talking about what's going on in the nucleus. In prophase, the chromosomes become visible. In prometaphase, the nuclear envelope disappears and the spindle fibers attached to the kinetochores. In metaphase, your chromosomes line up. In anaphase, the sister chromatids pull apart. And then in telophase, the nuclear envelope reforms and the DNA goes back to chromatin form. Cytokinesis is not part of mitosis, but it is occurring at the same time as mitosis. So remember, cytokinesis is just talking about how does the rest of the cell divide. What we'll often see is that cytokinesis may begin during anaphase or sometimes even during telophase, where you'll start seeing that cell pinching apart. Just remember that these are distinct events. Mitosis dividing the nucleus, cytokinesis dividing the cytoplasm. They do overlap, but they are distinct elements. Your textbook does a great job of diagramming this out. So here we can see our cell in interphase. The DNA is all in chromatin form. When we get to prophase, the DNA has now condensed. In our picture of prometaphase, you can see the nuclear envelope disappearing, and those spindle fibers are grabbing onto those kinetochore proteins. In metaphase, all the chromosomes have lined up on this halfway point. In anaphase, the sister chromatids begin to separate, and then in telophase, the nuclear envelope begins to reform. The DNA goes back into chromatin form. And this picture has a nice job of showing us how that cytokinesis is also occurring. You can see that the cell is starting to split apart. I do want to talk about the mitotic spindle and how it's helping this entire process to happen. The mitotic spindle is a bunch of microtubule structures that are helping these chromosomes move throughout the process. So remember, there's a region in our cell called the centrosome. This is an area where microtubules grow from. It's this yellow area over here. That's a centrosome. Not the stuff that's in there, just that yellow area. The centrosomes replicate during interphase, during G2, and they move to opposite ends of the cells during prophase and prometaphase. So that's part of the mitotic spindle, this centrosome. Also part of our mitotic spindle are these things called the spindle microtubules. The spindle microtubules are these little yellow spaghetti noodle looking things. And then also the third part of our mitotic spindle is the aster. The aster is this short array of microtubules that comes out of the centrosome. So when we talk about the mitotic spindle, it's those three elements, right? We've got the centrosome, we've got the spindle microtubules, and we've got the asters. In prometaphase, microtubules begin to attach to kinetochores, and they begin to move the chromosomes around. Notice that not all microtubules are attaching to kinetochores. For example, this guy right here and this guy right here. They haven't grabbed onto any microtubules, unlike over here and here. So some of your spindle fibers are grabbing onto the kinetochores. That's what happens during prometaphase. In metaphase, we're going to move everything around until we've lined up along an imaginary half-wave line called the metaphase plate. This is imaginary. There's not a piece of glass in the middle of your cell. At anaphase, the cohesins are cleaved by an enzyme called separase. So if we think back to our duplicated chromosome, right, here's our duplicated chromosome. And then we had that molecule in between them called cohesin that was holding them together. What happens in anaphase is that there's an enzyme called separase that comes and cuts the molecule in half that allows those sister chromatids to separate. The sister chromatids then separate and move along the kinetochore microtubule towards opposite ends of the cells. It might be really easy to think that this happens in the following method. So let's see if we can do it with my hand. Maybe this way. So it might, if my hand was a microtubule and my pencil here was a sister chromatid, it might make sense to your brain to think, oh, the microtubule grows out, it grabs onto the piece of DNA, and then it pulls the DNA to the opposite side of the cell. It's completely wrong. It's not the way that we want to think of it. Instead, the following happens. So you've got your chromosome out there. Let me try to keep it in the image for you. All right, your microtubule grows out and grabs onto that piece of DNA. Then what happens is, imagine that my fingers just fell off, and this poor piece of DNA is like, the fingers just fell off, run! And it starts running towards the other side. And then my hand falls off and it's like, keep running. And then my forearm falls off and it's like, keep running. And that piece of DNA is running towards the opposite end of the cell. How would we say this in science language? Okay, so we would say the following. The microtubule, my arm, right, those long things that are coming out and are grabbing onto the kinetochore protein. Let's add in our kinetochore protein too while we're at it. Those microtubule ends are shortening by depolymerizing. They're falling apart right here. That's what we see happening in this picture down here. As they fall apart, there are motor proteins. Remember from lecture? Those motor proteins are then moving that DNA molecule to the opposite end. So again, you should know this movement of how these chromosomes are sort of moving towards the opposite of the end of the cell. They're not being pulled. There are motor proteins that are walking them towards the opposite end. Non-kinetochore microtubules are still important. So these microtubules that are right here, they haven't grabbed onto any kinetochores. They haven't grabbed onto any chromosomes. They're still important because what they're doing is they're going to overlap and push against one another, elongating the cells. So again, looking at sort of my arms, I'll try to back up here a little bit. If you imagine this cell, and these are non-kinetochore microtubules, as they push against each other, my elbows are getting farther and farther and farther apart, right? It's stretching out the cell. The same thing is happening to us within our cell. And then in telophase, the genetically identical daughter nuclei form at opposite ends of the cell. We're done talking about mitosis, the division of the nucleus, so now we want to focus in on the cytokinesis, division of the cytoplasm. Cytokinesis in animal cells happens through a process called cleavage. Cleavage is the verb, it's the action, okay? The structure that we get is a structure called the cleavage furrow. The cleavage furrow is that sort of interesting wrinkle-looking picture that we've got in the corner. What happens in animal cells is that the two cells begin to pinch together, pinch, 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 pinch. That process of pinching is called cleavage, and that funny little indentation that you get, that's the cleavage furrow. Plant cells can't do this. They have a rigid cell wall, so you can't just pinch them in half. So they're going to need to do a process called cell plate formation. In this process, they're going to make a structure known as the cell plate. So here what we see is cytokinesis beginning to start. Mitosis is already finished. We have two new nuclei in the cell. And so what begins to happen is the cell begins to put down vesicles full of cell wall material. And they keep putting down more and more cell wall material until all of this cell wall material begins to form what's called the cell plate. That's why this process is called the cell plate formation. This process continues until the cell plate has divided that entire cell that used to just be one into two. Here are some visualizations of this happening in cells. So here you can see cleavage happening, and we can see the cleavage furrow. Whereas in the other cell, we can see the cell plate, and we would say that cell plate formation is happening. So this first one would be an animal cell, and the second one would be a plant cell. So this gets us sort of to the end of talking about the process of cell division and the cell cycle. So you should understand this picture pretty well, right? Again, if we're a brand new cell, we begin right at the beginning of interphase. This is where we spend our time mostly. As we go through interphase, we go through G1, S, and G2. After interphase, we then move through the M phase, which is composed of both mitosis and cytokinesis. And with mitosis, you should know the events of PPMAT. I'm never going to ask you to draw this out because that would be impossible online, but you should be able to describe or write out what happens during each of these phases. Don't only focus on PPMAT. It's important to be able to tell me what happens during S phase, or what happens during cytokinesis, or what happens during G2 phase. So you should understand and feel comfortable now with all of these processes that are related to the cell cycle. So I've got some questions for you to think about and see if you can answer them before I do. First question, what's division of the nucleus called? Hopefully that one wasn't too hard. Division of your nucleus, is mitosis. All right. Do prokaryotes have a nucleus? Hopefully, again, that wasn't too much of a hard one there. Do prokaryotes have a nucleus? No. All right, then here's my next question. Can prokaryotes, if they don't have a nucleus, can they do mitosis? The answer is no. Prokaryotes can't do mitosis. They don't have a nucleus, so they can't do this thing that's dividing the nucleus. Prokaryotes, bacteria and archaea, reproduce by a type of cell division known as binary fission. In this process, the chromosome will replicate, the two daughter chromosomes will move apart, one cell will get pinched in two cells. It's going to look really similar to mitosis, But we can't call it mitosis because there's no nucleus and there's no mitotic spindles or microtubules driving this process. So here we see our bacterial cell up in the corner. Our bacterial cell is getting ready to divide. So first thing it's going to do is going to replicate its DNA. So now you can see we've got double the amount of purple stuff in there. Those two pieces of DNA begin to move towards opposite ends of the cell. As they move towards opposite ends of the cell then begins to pinch apart and end up with two new daughter cells. Again, this process is called binary fission. This is how prokaryotes are able to go through the process of cell division. All right, let's see if we can do this without getting too confused. In this cell right here, how many chromosomes are there? Go ahead and just take a guess. In this cell right here, there is one chromosome. Again, remember that one duplicated chromosome, even though it has two sister chromatids in it, is considered one chromosome. Concept 12.3. Now that we know what the cell cycle system does, we want to look at how it's regulated by molecules within a molecular control system. Frequency of cell division really varies with the cell type. For instance, your skin cells are constantly dividing. However, your liver cells right now, they're not dividing. If you do get some damage to your liver though does have the ability to go through some cell division and regenerate in some fashion. Think about, however, about your brain cells, right? If you have a brain injury and you break or lose some of your brain cells, you can't grow them back. And so we want to think about, well, why is that? My skin cell, my brain cells, they have the exact same DNA in them. So why is it that my skin cells can divide all the time, but my nervous system cells are not dividing as often? And really the differences result from some sort of cellular regulation that's happening at the molecular level. The cell cycle is directed by what's called a cell cycle control system. This control system is looking at both internal and external signals. And there are specific checkpoints where the cell has to stop, just like at a border checkpoint, and has to show certain molecules or certain events before it's allowed to pass on. There are 3 checkpoints that we're going to talk about. We're going to talk about the G1 checkpoint, the G2 checkpoint, and the M checkpoint. Of these, the G1 checkpoint seems to be the most important, but there's also important signals that get looked for at the G2 checkpoint and at the M checkpoint. Again, these are places where the cell sort of has to stop and pause. It can't proceed unless it has some sort of signal. Let's begin by talking about the G2 checkpoint. I need you to understand how cyclins and cyclin-dependent kinases are used for a cell in order to get past the G2 checkpoint. So, the G2 checkpoint requires 2 elements. You are going to need a molecule called cyclin and a second molecule called cyclin-dependent kinase. If you have both, if you have cyclin and you have cyclin-dependent kinase, they combine to form a molecule called MPF. That stands for maturation promoting factor. This is a cyclin-dependent complex, kinase complex. And if you have MPF, this is how you get past the G2 checkpoint. Let's look at these molecules on the image and see if this can make better sense. Cyclin is the purple molecule in the picture. Okay, well, it's this molecule. We'll even draw it in the same shape. Cyclin, as the name suggests, cycles throughout the cell. Sometimes it's low, then it's high, then it's low, then it's high, and it's constantly in flux. It's cycling. There is not very much cyclin during the G1 and the S phases. However, when we get into the G2 phase, cyclin begins to accumulate, and we will have cyclin that's there during G2 and through the start of M phase. So cyclin is low during G1 and S, and then it starts building up during G2 and M. Then it goes down, then it goes up. There's a second molecule called cyclin-dependent kinase. It's the orange molecule. It's the square. Cyclin-dependent kinase is always there. It's just there waiting, waiting, waiting. What happens at the G2 checkpoint is that you need both of those molecules to come together. So we can see that happening in the image. We've got cyclin-dependent kinase and we've got cyclin. They come together to create that MPF. That MPF molecule, the cyclin plus the cyclin-dependent kinase, is what we need so that we can get past the G2 checkpoint. This is a way for the cell to ensure that the appropriate machinery has grown that's needed to in the G2 checkpoint before going into the M phase. The most important checkpoint seems to be the G1 checkpoint. The G1 checkpoint happens right near the end of G1. If a cell does not pass the G1 checkpoint for whatever reason, think something like this cell down here, it goes into a non-dividing state called G sub O. This is what happens to many of our nervous system cells. So if we think about our brain cells, early on during development, many of our nervous system cells hit to this G1 checkpoint and they didn't have the right signals. And so what happened is they got put into this G sub O non-dividing state. Unfortunately, what that means is if I dive into the shallow end of the swimming pool and I get some brain damage, right, my body is now unable to take these cells and put them back onto the cell cycle. Whereas something like a skin cells easily make it past this G1 checkpoint as they're able to divide very easily. The M checkpoint happens in between metaphase and anaphase. Cells will not begin anaphase until all chromosomes are properly attached to the spindle at the metaphase plate. So if we look at this first cell down here on the left-hand side, when the cell looks like that, right, so we've grabbed onto all the chromosomes, but they aren't quite lined up yet, and we're still moving around, this checkpoint's going to stay up and it's going to say, hey, we're not ready, we're not ready, we're not ready. It's going to wait until all of the chromosomes are aligned. Once all the chromosomes have been aligned, then this checkpoint will be allowed to be passed and we'll go into anaphase. So you've got those three checkpoints, G1, G2, M checkpoint. You should know that the G1 checkpoint is our most important checkpoint. You should understand how cyclin and cyclin-dependent kinase work to produce MPF for the G2 checkpoint, and you should understand how this M checkpoint works as well. Some other examples of signals that we will need to get past these checkpoints include things called growth factors. This is an external signal. These stimulate other cells to divide. One example of a growth factor is something called platelet-derived growth factor. If you take cells and try to grow them up in the lab, what will happen is they will be happy and they will sit there and they will do nothing. If you want cells to divide, you have to add an extra factor, something like platelet-derived growth factor, to tell them to divide. Some other examples are something called density-dependent inhibition. What happens with cells is cells will divide and divide and divide until they bump into a neighbor. As soon as they've bumped into a neighbor, they're going to stop dividing. So they're going to grow in one single layer. This is called density-dependent inhibition. Animal cells also exhibit something called anchorage dependence. This is where cells have to be attached to a substrate in order to divide. If the cell is just floating around, it won't divide. And now we want to start thinking about cancer cells. Cancer cells don't care about any of the rules that we've just set up. They don't care about having the right signals. They don't follow the cell cycle control system. They don't worry about this density-dependent inhibition or anchorage dependence. So here we can see on the left-hand side what a normal cell is supposed to do. A normal cell has to be anchored to something in order to divide. Once they get a single layer of cells, they'll stop dividing. If you remove a little bit, they'll quickly refill it. Whereas a cancer cell, if you look at those cancer cells, right, they're growing on top of one another. They're growing uncontrolled. They don't have that density-dependent inhibition. Cancer cells are not responding normally to the body's control mechanisms. One way that we've learned a lot about cancer cells is through a group of cells called the HeLa cells. HeLa cells stand for Henrietta Lacks. This is Henrietta Lacks over here. She was a woman who had cancer back in the 1950s, and scientists, without asking for permission at that time, removed from some of her cells and they started to try to grow them in the lab. The reason they took some of her cancer cells is that at that time we didn't have any human cell lines that could grow in the lab. The cells would live for a little bit and then they would die. And what they were looking for was some sort of human cells that they could grow up in the lab over and over and over again so they could have these cells to do research and understand biology better. For whatever reason, Henrietta Lacks cells did amazing. They were still alive a week later, 2 weeks later. As long as you divided them and put them into a new container with enough food in them, they kept growing and growing and growing. And so the scientists got really excited. They started calling up all their other scientist friends, and these cells were sent around to all various types of labs across the United States and across the entire world. To protect her, these cells were called HeLa cells. And many people only ever knew these cells as HeLa cells. They knew nothing about Henrietta Lacks. She unfortunately passed away from the cancer. It was a very aggressive form. She did not survive, but she did have a family. She had children who subsequently had grandchildren, and their family knew nothing about this. These cells have been really important. These cells are still alive today, so they're still being used today. Again, it's because these cells are cancer cells. So what they're doing is they're making their own growth factor, or they might be telling the cells to divide without a growth factor being there, or they might have an abnormal cell cycle control system. This book over here, or now made into a movie by Oprah, maybe you've seen that version of it, is not a science book. I would encourage you to read it if you're looking for something to fill some of your time while you're spending more time at home, or watch the Oprah movie. This is a story of this entire process and really talking to this lady's family and her descendants and figuring out what's going on. Unfortunately, these cells are worth millions of dollars. There's patents based on these cells. They've been the basis of winning Nobel Prizes, and her family has never seen any of this money, has never received any recognition for her involvement in this entire process. So the book is just a really great look at what went on. This is an example where scientists really got it wrong ethically. Transformation is when a normal cell gets converted to a cancerous cell. So unlike many other diseases, cancer isn't caused by something external coming into the organism. Instead, cancer is caused when your own body is breaking in some fashion. Cancer cells usually are eliminated by our immune system. So most of the time, our own body is watching for cells that have broken, and it will eliminate it long before we ever recognize that something was wrong. When our immune system can't keep up with these abnormal cells, these cells may form tumors. Tumors are masses of abnormal cells within otherwise normal tissue. A benign tumor is pretty common. You shouldn't freak out if you know that you have a benign tumor or someone else has a benign tumor that you care for. This is where the original cells remain at their original site. So think something like a wart or a little lump in your skin. As long as it's not getting bigger or it's hurting surrounding areas, oftentimes doctors will just let it stay there. If, however, that tumor begins to get bigger, doctors will remove it because they are worried that that tumor can become a malignant tumor. A malignant tumor is one that can invade surrounding tissues. Worst case scenario, a malignant tumor metastasizes. That's when a chunk of the tumor breaks off and then it moves throughout the organism and it lands someplace else. So here we can see we've got a breast tumor. Here it is becoming a malignant tumor. It's starting to invade surrounding tissues. And then worst case scenario, it's metastasized. A little piece of that tumor has broken off. It's gotten in our blood and our lymph system, and now it's moving and it's causing cancer elsewhere in the body. Notice that these metastatic cancerous cells are no longer responding to that rule that cells have to be stuck on something to have that anchorage dependency in order to divide. What are the treatment options for somebody who has cancer? You could do radiation for localized tumors. That's going to damage the DNA in the cancer cells. Unfortunately, it does often cause a lot of damage to normal, healthy cells. So there are a lot of painful side effects. Chemotherapy is where doctors will prescribe certain poisons. The idea is that it will poison the cancer before it poisons the patient. It's targeting cells that are going through a rapid cell cycle. So some of the side effects that you'll get is things like your hair falling out, right? Because those skin cells, those hair cells are often dividing really rapidly. Or your stomach will get really nauseous because those stomach cells are often dividing really rapidly. So chemotherapy, yes, will be able to target those cancer dividing cells, but it will also attack any rapidly dividing cells. There are also personalized treatments. These are more expensive, where you might be able to teach your immune system how to attack your own cancers. What we want to do now is we want to jump to chapter 18.5, pages 386 through 392, and talk about some genetic changes that can affect the cell cycle control system. So in chapter 18.5, we're not covering all of it. We're just doing this little teeny bit to look at cancer. Cancer is often caused by mutations to genes that regulate cell growth and division. Proto-oncogenes are genes that we all have in our body right now. They're good. We want them. One specific example of a proto-oncogene is a gene called a RAS gene. What happens, unfortunately, in some individuals is their proto-oncogenes get mutated or broken and they turn into oncogenes. Oncogenes are bad. Those cause cancer. So that's one class of proteins. The other class of proteins are called tumor suppressor genes. These tumor suppressor genes are, again, a good thing. You have these genes in your body right now. They're suppressing tumors. That's good. One example of a tumor suppressor gene in your body right now is P53. The problem that happens in cancer is sometimes these break. If your tumor suppressor gene breaks, if P53 is mutated, then this is going to lead to uncontrolled cell growth. So make sure that you understand sort of these two basic types of genes. We've got proto-oncogenes and tumor suppressor genes. And then additionally, make sure you know examples of each. RAS is an example of a proto-oncogene. P53 is an example of tumor suppressor genes. And again, these are good genes. We want to have active RAS in our body. We want to have active P53 in our body. The problem that occurs is when these genes get mutated. So we'll go through each instance. Let's start with RAS mutations, starting by looking at the normal top picture. In the first picture, there is a growth factor that's binding to this cell. So there's a signal that says, hey, cell, you should divide. This turns on the RAS gene, so the RAS protein is activated. This results in the cell then dividing. That makes sense. There was a signal that said cell divide, RAS got turned on, the cell divided, right? That's what's supposed to happen. Let's see what happens in somebody who has an oncogene, somebody who's had this gene mutated. So note, there is no external signal telling the cell to divide. There's no reason for the cell to divide. However, RAS has been mutated. RAS is hyperactive. So RAS on its own starts telling the cell divide. And it keeps telling the cell divide, 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 and this leads to an increase in cell division. So let's look at the tumor suppressor gene. Let's talk about P53. So again, we'll talk about what's supposed to normally happen in a healthy scenario. If you go to the tanning salon and you expose your body to UV light, you are causing DNA damage. Stop it. Don't ever go to the tanning salon ever again. It's so dangerous for your cells. So what happens is the UV light comes in and it damages your DNA. Your body is smart, so your body recognizes that these cells are potentially problematic because they've got DNA damage. So what happens in these cells where there's DNA damage is through a cell signaling pathway, this will activate P53. P53 is supposed to prevent tumors, and so what happens, it says, stop, don't divide. And so this cell will no longer divide, which is good, right? You've damaged this cell, it's broken in some sort of way. P53 says stop, let's not divide the cell anymore. See what happens in somebody who might be developing cancer. They've damaged their DNA in some sort of way, right? There's some sort of DNA damage, but in this case, P53 has been mutated. It's missing, it's no longer there. And so what happens is this cell is going to be allowed to divide over and over again, even though there is damage to the cell. So we can sort of see two different things that happened. When we looked at our oncogene or the proto-oncogene, what we saw is that the cell cycle got overstimulated. With the RAS oncogene, RAS said on, divide, 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 which led to lots of cell division. On the other hand, our tumor suppressor protein, when we looked at P53, it was broken, right? It wasn't hyperactive, it was missing. And but that again resulted in divide, 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 and both of those can lead to cancers. You have a great Science Friday video about naked mole rats. I encourage you to watch that. In order to develop cancer, many mutations are generally needed. Cancer levels are going to increase with age. Simplistically, maybe think of it like this. In order to get cancer, you need to get 10 mutations, right? So you're born and you have no mutations in your DNA, and then you're 1 year old, you're 2 years old, you're 3 years old, you're 10 years old, you're 15, you're 20, you're 40, you're 50, you're 60, you're 70, you're 80. Now you've got cancer. Right? Because you've been around for so long that you've had more opportunity for all of those mutations to accumulate. Cancer, usually what we see is there's at least usually one active oncogene and multiple tumor suppressor genes. Down across the bottom, you can see an example of colorectal cancer. Here's an individual that has a normal, healthy colon, right? Everything's nice and smooth. When the genes start to mutate, you may start to get small benign growths. So this person doesn't have cancer yet, right? They've just got this benign tumor. This is why your parents should go get their colonoscopies even though they think it's gross. This is why you should listen to your doctor when he says it's time to go get his colonoscopy. It's really easy to see these groups of cells that are growing out of normal, and it's really easy at this stage to just remove them, right? And then this person would not have to worry about those cells getting any further mutations. If, however, the person does not get their colonoscopy and doesn't recognize that they have these cells that are starting to be broken, that start to have mutations, additional mutations can begin to develop. And as those additional mutations develop, we get larger and larger growths, and eventually with the additional mutations that happen, we can then get the full-blown cancer where it's invading surrounding tissue and causing quite a lot of damage to the individual. When we speak about cancer, I want you to know that cancer is a heterogeneous disease, meaning that individuals can both say that they have lung cancer, but they can have very, very different mutations that gave them lung cancer. This is an example that your textbook gives of breast cancers. You do not need to memorize all these subtypes. I just am using this as a moment to look at how breast cancer can vary differently from one organism or one individual to another. You've probably seen this in your own life. You probably know someone who battled breast cancer and is now completely okay and healthy. And then you probably know someone who has battled breast cancer and they passed away 6 months after. And you are trying to figure out, well, why did it work for one person and it didn't work for the other person? It turns out with breast cancer, we call all of these breast cancers, there's actually 4 different types. So let's start with the picture in the upper corner. This is what a normal, healthy breast cell should look like. There are some estrogen receptors, those are the orange pieces or the pink pieces. There are some progesterone receptors, those are the green pieces. And then there's these HER2 receptors, those purple things on the outside. That's what a normal, healthy breast cell is supposed to look like. So you can see, as we look over on the other side, what might happen in some different types of breast cancer. So if we look in this upper picture right here, luminal A, these individuals, they have a lot more oranges, they have a lot more greens, and then they're missing that HER2. And people who have this type of breast cancer, it's still bad, it's still not great, but these individuals are often able to fight it off and survive. Versus somebody like this who has basal-like. They're missing all the orange pieces, all the green pieces, and the HER2, and this is really aggressive, right? Much less likely that you'll be able to survive. So again, what's the take-home message from here? Is to recognize that when we talk about cancers, it's a really big, broad, generic term that every individual is going to have their own unique mutations that are contributing to that cancer. And this is the one reason that cancer treatments are sometimes going to be better for some individuals and not as efficacious for others. Individuals can inherit predispositions and other factors that contribute to cancer. You can inherit oncogenes or mutant alleles of tumor suppressor genes. In colorectal cancer, you can inherit a gene called adenomatosis polyposis coli. You can also inherit a gene called BRCA1 or BRCA1 or BRCA2 for breast cancers. If your grandpa has skin cancer, It is unlikely that you inherited a skin cancer gene from your grandpa. What we're saying is before you were even born, if your parents had a gene that was in their egg and sperm, they could have passed that on to you. So when you were born, instead of being born with no mutations, you might have already been born with a couple mutations. Does that mean that you're going to get cancer? No. Maybe you're 10, you're 20, you're 30, you're 40, you're 50, you're 60, you're 70, you're 80, you're 100, and you never get enough mutations to cause cancer. What it does mean, though, is that you've already started off with some of those mutations. So somebody like Angelina Jolie, whose mother passed away from breast cancer, she tested and found out that she was BRCA1, BRCA2 positive. She did not have breast cancer, but because she was concerned, she went through the radical mastectomy to remove that breast tissue because she didn't want to worry about it. So there are all of those tests. Just to think about these inherited cancers, less than half of the cancers are a familial or an inherited cancer. Most cancers are just unfortunately random mutations that occur. If you have a family history of cancer, yes, I would strongly encourage you to speak to your doctor and consider getting genetic testing for those genes that you may have inherited. If, however, you're just generally worried or you want to know about cancers, these genetic tests probably aren't worth your money because, again, most cancers are happening just as random mutations. Viruses can also cause cancer. Some tumor viruses cause cancer. So if we think about a virus, right, a virus infects the cell, it comes in and then it inserts its DNA into a particular location. Sometimes when viruses do that, they put their viral DNA in exactly the wrong place in our cells, and that can cause cancer. One example is HPV, human papillomavirus. 1 in 4 people have human papillomavirus infections. It's just a very common infection. But for some people who have been infected with that human papillomavirus, that viral DNA is inserted in just the wrong place and it causes cancer. Almost all cases of cervical cancer, when we look at the genetic component of what's causing that cancer, we find out it's this HPV virus. And so that's why there's a vaccine against HPV to hopefully prevent this cervical cancer. This gets us to the end of chapter 12 and chapter 18. If you have questions, please hop on to Zoom, ask me some questions, send emails, and as always, happy studying, and I look forward to seeing you soon.